Hi everyone, this is part 2 of the genome expression lecture. I'm going to finish off the translation part. We have already talked about the replication and transcription. We're going to finish off the translation within this part. So we've talked about the genetic code already. We've talked about how the DNA has these regions that we define as gene, and we talked about the structure of the gene extending from promoter to the terminator, and then the transcript having leader and terminator sequences as well, or I believe it's leader and trailer sequences. We're going to today talk about how that transcript in form of mRNA will be used to synthesize protein. So DNA had two strands, a template strand that is used to synthesize the message, right? And we talked about that there could be different types of RNA coding genes. You could have genes that code for tRNA, or genes that code for ribosomal RNA, and then genes that code for the messenger RNA. And it's the messenger RNA that serves as a message for synthesis of proteins. So we're going to be discussing the further processing of that mRNA into protein. But here, I want you to focus a little bit on the structure of the mRNA. And notice that the sequence of the RNA is identical to one of the two strands of the DNA. In this case, you can compare it to the two strands, and you notice that it's exactly the same as the coding strand, except for in place of the thymines, you have the uracils, because RNA does not have thymine, of course. So the top strand in this case is the coding strand for this gene, and the bottom strand was used as a template against which this mRNA was synthesized using the RNA polymerase. And when the RNA is synthesized, a portion of it is going to be referred to as the untranslated region, and you're going to have one in both the 5' end and the 3' end of this mRNA. And we're going to talk about the significance of it in a moment. But just downstream to that is going to be sequence that will be converted into proteins, and that sequence is read in triplets, and we refer to these triplets as codons. And generally, you would have a specific codon that is found at the beginning of this protein coding region, and we refer to that triplet as the start codon, and it often happens to be AUG. It's not always AUG, but for majority of the proteins, it happens to be AUG. So this is where the ribosome will begin to recognize the beginning of the protein, the first amino acid. It will learn to read this triplet as the first word of this sentence, and then it will continue to read nucleotides beyond this in triplets as well. So it knows exactly where to start reading and then how to kind of group them into groups of 3. So we refer to these groups of 3 as codons, and there's going to be multiple codons as we're going to look at in a moment. Now, each of these codons have to be recognized by the ribosome to mean a particular amino acid. And so these codons are recognized by anticodons that are present on the tRNA. So hopefully you've been kind of keeping track of all the RNA that are involved. So messenger RNA was serving as the message. Ribosomal RNA, which is part of the ribosome, would be involved in this translation process of converting this mRNA into protein. And then the anticodons that belong to the tRNA or the transfer RNA are essentially responsible for ensuring that there is this correct match between the codon and the associated amino acid. Okay, so for example, the start codon AUG corresponds to a tRNA that has the anticodon that base pairs in a complementary fashion to AUG. So it has UAC as the anticodon. And once that match has occurred, then methionine is added to the growing polypeptide chain. Okay, so this genetic gene to protein connection involves all three types of RNA molecules, as we're going to see. And it's only the mRNA that is converted into proteins. Okay. So when we look at the protein structure, it's comprised of 20 different amino acids. And these amino acids have a unique sort of basic structure to them, that there is a central carbon that we refer to as the alpha carbon, and it has four moieties attached to it, just like any other carbon. Three of those groups are identical in all amino acids, and those would include an amino group, a carboxyl group, and a hydrogen. And the fourth group is referred to as the R group, and this is the variable group. This is where the different amino acids differ from one another. Now, there are many different varieties of amino acids, but generally there are 20 that are found within proteins that are synthesized by the ribosome. And then there are other amino acids that might be incorporated into proteins, or existing amino acids might be modified into pre-synthesized proteins. Okay, but so there are these 20 different amino acids at least that are going to be attached to, at least 20 different tRNA molecules. There's going to be more, as we're going to see, because there are many different codons. But so you're going to have at least, 20 of these, and these are going to be linked together via peptide bonds on the surface of the ribosome into a polypeptide. Okay, so protein has a directionality the same way RNA has it, DNA has it. They're 5' to 3'. In this case, protein is generally synthesized from amino terminal to carboxyl terminal. Okay, and so you have basically the carboxyl group of the first amino acid react with the amine group of the incoming amino acid. Okay, so we can look at multiple peptide bonds in this small polypeptide that we're seeing. Here you have the amine group, and this is the alpha carbon, and this is the carboxyl group that has reacted with the amine group of the second amino acid here. Okay, so this is one peptide bond, and you see it in second peptide bond. There's a third peptide bond. Okay, so you're going to have these multiple amino acids that will be linked together on the surface of the ribosome via these peptide bonds. And then once you have this primary structure of the protein, which is simply just a chain of amino acids, it will fold into its secondary structure and the tertiary structure, and in its tertiary structure, it will be ready to interact with either other proteins or other polypeptides, and it will be functional only in its properly folded form. Okay, but once protein is synthesized, synthesis, and it's being extruded from the ribosome, you're going to see the amino end first, and the carboxyl end is the last one to be synthesized. So the first amino acid, the amino group, would be the one that corresponds to the first codon, and the last amino acid next to the carboxyl group is going to be the last amino acid coded by the last codon on that particular mRNA. Now, when we look at the number of codons, there are 4 different bases, and you can combine them in 64 different ways. 4 raised to the power 3 is going to give you the number of different combinations that are possible using 3 bases to synthesize a codon or to make up a codon. And so when we look at the genetic code, we're basically looking at all the different combinations that are possible for using, the 4 codons, 4 bases rather, that we have: the A, the C, the G, and the U. And so there is an easier way to read it. You could quickly go through all the codons. Let's say you're looking for, a AAA, you kind of quickly go through it and you find AAA, and you notice that next to it, it says lysine, so it codes for lysine. But there's an easier way. There's a method to this. You first look at the left side of this chart and you find A, and then you go across and you find the second A, and then you go down and you find the third A, and you will again come to that AAA, the same site. So that's just how to look for it. You could do it any way you like. Idea is that you should be able to look at it, a triplet, a codon, and tell us what the corresponding amino acid might be for that particular codon. One of the things that I want you to notice is that the code is redundant. Remember, you have 20 amino acids that these codons are coding for, yet you have these 64 different codons. And so you obviously don't have 64 different amino acids, and these codons aren't necessarily represented equally in the genetic code within different organisms. So you may find that some organisms have a preference for a particular code. That's just how they have evolved. And so we do see what we refer to as codon bias among organisms. And so I want you to notice that there are some amino acids that are encoded by several codons. So leucine, for example, here is encoded by 6 different codons. Okay, so your genetic code and the genetic code of these microbes is redundant, which means that several codons are going to be coding for the same amino acid. That's not true for all of them. Methionine is encoded by a single codon, so AUG only encodes for methionine, but then you have things like threonine and alanine and valine. Each one of them have 4 codons. And one of the things that you want to keep in mind is that often when there are multiple codons for a particular amino acids, often the first 2 bases happen to be the same as the third base where they vary from one another. So that's, ideal to basically ignore mutations that might happen at the third position on the codon, right? So there are sometimes mistakes that will be made during the replication process, very rarely during the transcription process as well, but as long as the first 2 bases in the codon are the same, the third base doesn't really matter, okay? So we're gonna see that this makes the genetic code a little bit forgiving, and that you could have errors and it not really impact the overall protein structure. So that brings us to the concept of tRNA wobble. So although we have 64 different codons here, there is one that codes for the initiation codon, which is the first codon of any given protein. And then you have 3 separate codons that don't code for anything. Okay, so we refer to them as stop codons because they serve as a signal for the ribosome to stop synthesizing further protein. Okay, so they will not add any additional amino acid once they arrive at that stop codon, and they will get ready to disassemble. And methionine is going to be that first amino acid, as I mentioned earlier, the initiation codon, that it's going to code for a amino acid, right? So the two differ in that initiation codon actually codes for an amino acid, and it's methionine, whereas the stop codons do not code for an amino acid. So there's 3 stop codons, and there are 63 separate codons that code for amino acids. Do we have 63 tRNA molecules to correspond to that? And what we notice is that that's not the case. One, because as I mentioned earlier, that some genomes have certain preference for some codes, right? So that it prefers maybe 2 out of 4 codons for, say, valine, all right? So it would just have maybe 2 tRNA molecules corresponding to that. The other thing that is important is that if we look back to valine, the first 2 bases are the same among those 4 codons, and it's just the last base that differs. And so what we notice in tRNA molecules is that there is somewhat of sort of lax base pairing that is allowed at that 3' position of the codon. So in this case, we're looking at codons for glycine. There are 2 separate codons, GGU and GGC. And if you were to have this ideal situation where there was a individual tRNA molecule for each of the 2 codons, what you would find is that you would have one tRNA that would have, CCG as the anticodon, right? And the other one that should have CCA as the anticodon, right? So CCG will correspond to GGC, and CCA would correspond to GGA, but that's not GGU rather. But that's not what we notice. Okay, what we notice is that there is one actually tRNA that is able to correspond to both of these codons, meaning it's able to recognize both of these codons in the mRNA as the code to insert a glycine into the growing polypeptide chain. And so this is allowed because there is this misbase pairing that is allowed at the 3' position of the codon. So as long as the first 2 bases are CC, these 2 codons that you're seeing will be recognized as, those coding for glycine, and you would have the same tRNA recognize both of them. So it allows cells to have fewer tRNA molecules to correspond to greater number of codons. So you can have in bacteria 4 different codons that code for glycine in mRNA, yet there is only, 3 different anticodons here that correspond to those 4, mRNA codons. And so it doesn't always have to be this way. You can have all 4 codons be recognized depending upon the amino acid that we have and depending upon, of course, the genome of the organism, or you could have just one. Okay, so it varies from organism to organism and depends on the genetic code. Okay, so there is some sort of a tolerance that is allowed at the 3' position of the codon. Okay, so that would be actually the 5' position of the anticodon where G is highlighted, right? The 2 would have to base pair in complementary fashion and generally in, of course, the anti-parallel direction. Okay, so there are some exceptions to the code. Generally, we stick to the genetic code that we have already seen, but we do see some deviation from that code. For example, if we look at the mammalian mitochondria, the codons AGA and AGG signify stop. Okay, so that is a stop for the mitochondrial ribosomes to further add any additional amino acids. But if we look at normal organisms, AGA and AGG actually code for arginine. Okay, so there is some variation that you are definitely going to see. There are other organisms that can take stop codons such as UGA, okay, and they can insert amino acids in their place. Okay, so UAG, UGA, both are actually, they signify stop codon for majority of the organisms. But in some methanogens, for example, it's assigned to insert a pyrrolysine, which is a modified lysine, or UGA can sometimes signify addition of seleноcysteine, which is a serine modified amino acid. Okay, so you will see some deviations from that genetic code, especially in microbes because you have such variety. Now, when we look at some of these modified amino acids, as I mentioned, seleноcysteine is modified from serine and pyrrolysine is modified from lysine. Generally, they are going to be inserted in response to presence of a particular type of a stop codon. So it has to be, for example, UGA, right? It can't be, UAG or UAA. You would have to have UGA. And then there often happens to be a sequence next to that stop sign that actually lets the ribosome know that this modified amino acid would be inserted. So if you change any of those two parameters where either the stop codon is different or the insertion sequences have been changed, you will not have the insertion of these modified amino acids. Okay, so this is important for both insertion of selenocysteine. It has been demonstrated in presence of pyrrolysine being inserted into certain proteins. So, there is still a lot to be learned. Perhaps there are additional sequences found in other organisms that allow, for example, mycoplasma to recognize UGA as a codon for glutamine instead of just a stop for ribosome. So there will be exceptions to the code. Now, in terms of bacteria, they are unique in that they don't really have a nuclear membrane that separates their genetic material from the cytosol. Cytosol is where you would have generally ribosomes. And they have this nucleoid region, which is isolated somewhat from, of course, the rest of the cytosol where all the other enzymatic reactions are occurring, but it's not physically separate. So there's a lot of reactions that can occur concurrently. So transcription and translation can occur at the same time within bacteria. Generally within eukaryotic cells, the transcription would have to be completed, in eukaryotic cell within the nucleus, then the processed messenger RNA would have to be located into the cytosol or within the rough ER, and that is where it will be further, translated. Now, within bacteria, as soon as the RNA polymerase begins to synthesize the mRNA, it can serve as a site for the assembly of ribosomes. As long as the leader sequence has been synthesized, which has the signal for binding of the ribosome, then ribosome assembly can begin. And once that signal sequence has been already utilized by one of the ribosome has already gone past it, then you can have addition of further ribosomes onto, that particular sequence. So you can have multiple ribosomes that begin to synthesize protein using this message all at once. Okay, so we refer to this as the polyribosome complex, and you can notice that the ribosomes that are closer to the beginning of the mRNA, this is where the leader sequence is, this is where the initiation codon is. They're synthesized, they are synthesizing proteins, but the size of the polypeptide is actually shorter, and it makes sense because they've covered less area in terms of codons, and those that are much closer to the RNA polymerase or closer towards the end of the actual message, they have synthesized a much longer protein because they've read many more codons. So this is something unique that only happens in prokaryotic cells because they lack a nuclear membrane, and transcription and translation can occur together. And we refer to this phenomenon as co-translation. And this allows microbes to adapt very quickly to their changing environment. So they don't have to wait for all of this processing of mRNA to occur because there are no introns, right? We have introns and exons within eukaryotic genes, but not within the prokaryotic. There's a lack of introns. So as the mRNA is being synthesized, it doesn't need any further modification before you could translate it. So translation can begin immediately, and you can have multiple polypeptides synthesized, very quickly, right? Their synthesis at least begin even before the transcript has been fully synthesized. And so multiple of these proteins would be ready within a minute or so of this RNA polymerase having fully synthesized the mRNA. So it allows microbes to synthesize several copies of this protein simultaneously and just very quickly to adapt to the changing needs. So tRNA is a sort of a unique RNA in that it is able to fold back on itself to produce this clover leaf-like molecule, which has these different arms, the D arm and the I believe it's a Phi, but this is the TC arm. And these arms all have these sort of unique bases, and that's where their names are derived from. But there are some sort of bases that are conserved, so you always find them at particular sites, so they're represented as diamonds. It doesn't matter exactly, right now for our purposes, the actual structure of the tRNA, but of course these bases are important in order for it to properly fold into this, three-dimensional shape. The most important arms for translation purpose are the anticodon arm and the acceptor end, which happens to be at the 3' end. So anticodon arm, as we've already seen, it's going to have those three bases that are going to base pair with the codon in a complementary fashion. So A against U, G against C, right? So we're already aware of that. And then at the 3' end, there is the amino acid attaching site. Okay, generally these bases are conserved, so you would always have CCA at the 3' end, and the adenine is going to associate with the amino acid. And so this binding or the attachment of the amino acid to that adenine is carried out by various amino acid or tRNA synthases. Their job is to correctly recognize the amino acid that corresponds to this codon anticodon pair, right? And attach that at the 3' end. You could easily see that absence of such amino acid tRNA synthesis could be very problematic for these protein synthesis process, because if you don't have the amino acid tRNA synthase, that it doesn't attach the proper amino acid to a tRNA, and that tRNA can obviously take part in the translation process using its anticodon, but it doesn't have any amino acids to contribute to the process, right? So you're going to have at least 20 different types of amino acid tRNA synthases whose job is to attach the correct amino acid to the acceptor end of a tRNA molecule. In reality, there's going to be many more than 20. Okay, but you can imagine, since there are at least 20 different, essential amino acids, then you're going to at least need 20 different kinds. And so here you're looking at a simplified image of the same tRNA. Here you're seeing the C, additional C, and then this is the A of this acceptor end. And the amino acid, the correct amino acid, is attached to the 3' hydroxyl of adenine using a ester bond. Okay, and this happens to be a high energy bond, and this is going to require energy. So the amino acid tRNA synthase is going to use amino acid, tRNA, and ATP as a substrate to generate this, ester linkage of amino acid using its carboxyl group to the 3' hydroxyl group. Okay, so this is high energy bond. So when you hydrolyze this bond, it releases the energy for the synthesis of the peptide bond within the ribosome. Okay, so we're going to see that that particular step where the peptide bond is forming doesn't require additional energy, because as you hydrolyze this particular high energy bond, it's going to provide that energy needed to drive that next reaction, which is the peptide bond formation. Okay, so tRNA is folded and it has this, T-like shape or clover leaf-like shape. Now, the final type of RNA that we need to talk about is found within the ribosome. And we have looked at bacterial ribosome already. We know that there is a small subunit. We know that there is a large subunit. And both of them are part protein and part RNA, right? So if we look at the 30S subunit, it contains a 16S ribosomal RNA, which is going to be important because it's going to help orient the ribosome around the mRNA. It's going to help the ribosome sit accurately on the mRNA so that it knows where to begin to read the first codon, right? So it helps it establish the reading frame. And it has 21 different proteins, and similarly, 50S ribosomal subunit includes two small ribosomal RNA molecules, the 23S and the 5S, and 34 different polypeptides. So that's what this image is showing you, but I find it easier to describe the various binding sites using the simplified image, that when you have these two ribosomal subunit come together, they're going to together form these three regions that will allow the tRNA to interact with the rest of the machinery. And you have the A site, which is generally for the incoming tRNA, okay? The P site, this is also occupied by a tRNA, but this would be the tRNA that will be attached to the growing polypeptide chain. And then you would have the E site, this is for the tRNA to exit. So A is to enter, and then the same tRNA would eventually move into the P site, and then finally from P sites into the A site, and then it would leave via this little exit that would be present within the large subunit. Okay, so in order for these three sites to sort of complete themselves, the two subunits have to come together. Okay, and we're going to have within the larger subunit the 23S ribosomal RNA, which is going to act as a ribozyme for the synthesis of the polypeptide, and so for the peptide bond formation, essentially. Right, so the enzyme that is carrying out the reaction of linking two amino acids together is actually a ribosomal RNA, okay, which is the 23S. So we refer to it as the peptidyl transferase. Okay, so keep in mind there are going to be these three sites. The incoming tRNA will come in and recognize the codon in the A site, and then you would have one that would be sitting in the P site, and it would have moved there after it had first arrived to the A site, and then eventually when its job is done, it's going to leave via the exit site. So there are going to be three different codons that will correspond to these A, P, and E site, right? So E site codon would have been read first, then the P site codon, and A site codon is the one that is being currently deciphered, okay? So here's a video for you to watch that walks you through the translation process. It's a very brief video. Make sure that you watch it. Now, in terms of translation, the process occurs in three separate steps. You have the initiation, elongation, and the termination. The initiation step requires that you have the two subunits come together around the mRNA to fully contribute to the formation of the A, P, and E site. And it's important that you have the correct codons correspond to, those sites. And so in order to accomplish that, you're going to have the leader sequence associate with the small ribosomal subunit. So there is the 16S RNA in the small ribosomal subunit that will correspond to a sequence in the leader sequence of the mRNA that we refer to as the Shine-Delgarno sequence. Okay, so the 16S RNA of the small subunit is going to recognize the Shine-Delgarno sequence within the leader sequence of the mRNA, and it's going to help the small subunit sit along the mRNA, right? It will allow the two to base pair with one another. And then it will have the first codon sit in the P site, okay? Or what would eventually be completed into the P site once the large subunit associates with it. So a couple of things before that could happen. There are going to be obviously the two subunits. There's going to be the tRNA, there's going to be the mRNA, but there are initiation factors that will be referred to as IFs that would help with the initiation of the translation. So initially, when small subunit isn't involved in the translation process, it's found separate from the large subunit. And so that is accomplished by the initiation factor 3. It will bind to the small subunit and will prevent it from interacting with the large subunit until this initiation process has been completed. So that 30S subunit, once it associates with the mRNA along the Shine-Delgarno sequence, will be ready for the, of course, binding of the large subunit. But so that's one step, that you would have the 30S subunit associate with the mRNA in a correct fashion. But you also have to bring in the first tRNA that has a code anticodon corresponding to the first codon. And you'd remember first codon is the initiation codon, which always codes for AUG. So you're going to have the methionine anticodon or tRNA corresponding to methionine also be part of this complex. All right, so this initiation complex is comprised of the small subunit of the ribosome, it's comprised of mRNA, and it's comprised of the methionine carrying tRNA. Now, bringing the correct tRNA molecule to this complex is an energy requiring step, okay? Cells spend a lot of GTP ensuring that the correct tRNA will enter these various sites to ensure the fidelity of this process, so that only the correct amino acid would be added, and also to make it, sort of expedited in a way, okay, so that it's very efficient. You're not kind of waiting around to see, whether the right tRNA with the right anticodon would enter the A or the A site. So you really want the correct tRNA to enter the A site from the get-go, okay? And so there are going to be initiation factors whose job is to recognize that first tRNA. In this case, you're seeing the initiation factor 2, which corresponds to this formal methionine. It also binds GTP. And so it's going to recruit the tRNA, bring it to this complex, and initiation factor 1 is required to kick off initiation factor 3 from the complex. So initiation factor 3 is replaced by initiation factor 1. Initiation factor 2 is required to correctly sit this methionine carrying tRNA and onto the first codon. And then you would have the completion of this initiation complex by allowing the association of the large subunit. And once the large subunit associates with it, notice now the 3 sites have been completed. You have the A site, the P site, and the E site. And in the process, the initiation factors have also been removed, right? So this is now a complete unit that can begin the elongation of the synthesis. And so I want you to bear in mind that the first tRNA molecule, right, it's bound corresponding to the first codon that is sitting in the P site, but the second codon and every subsequent codon will be found within the A site, and so the corresponding tRNA molecules will first enter to the A site. Now, one of the things that I want to mention is the identity of the methionine that is added at the very beginning of the protein versus other methionines that are added at, say, maybe the middle of the protein, okay? So in bacteria, the first methionine is referred to as the formal methionine, and it has this formal group attached to it as shown in blue. And it prevents this amine group from taking part in formation of a peptide bond, right? In order for this to take part in a peptide bond formation reaction, it had to be NH2 so that it could react with the hydroxyl group on the carboxyl group of the previous amino acid. So this N-formal methionine, as we refer to it, is only inserted at the very beginning of the protein, because in the middle, the amine group is not really available to take part in peptide bond formation reaction. Okay, and once the protein has been synthesized, bacteria may elect to, remove that formal group, or even just remove the methionine altogether. But nonetheless, the initiation complex is complete now. We went from 30S and 50S subunits to now a 70S completed initiation complex. And the next step is that you're going to have the amino acyl tRNA enter into the A site. And again, this is an energy requiring step, just like we had with the initiation complex. We had the IF2, for example, recruiting the correct tRNA corresponding to the first codon. So we're going to have these additional elongation factors that are going to be represented as EF. So you're no longer going to see them being represented as IF, because I is for initiation and E is for elongation. So now these will be referred to as the elongation factors or EF factors. So you have a couple of EF factors here. Here you have the EFTU, who's responsible for using GTP and recruiting the correct tRNA that corresponds to the next codon. And once this codon matches, it hydrolyzes its GTP to GDP, leaves the tRNA molecule in place, and is going to replace its bound GDP for GTP. So it has additional elongation factor here, elongation factor TS, that is going to help elongation factor TU replace its GDP for GTP so that it could be recycled and it could bring in, additional tRNA molecules. But once that tRNA is in the A site, then you're going to have the peptidyltransferase, which is the role of 23S ribosomal RNA within the large subunit, undertake the peptide bond formation reaction. Okay, and once that peptide bond forms, then you're going to have the peptide that was originally in the P site for the moment be transferred to the tRNA that happens to be in the A site. Okay, so right now the polypeptide is within the A site. We need to bring in the next codon so that we could, of course, add the amino acid corresponding to the next codon to the polypeptide chain. And so we're going to have this step that we refer to as translocation step. Okay, and within the translocation step, the whole ribosome as a complex is going to slide down the mRNA one codon at a time. Okay, and so this process requires its own elongation factor. So you're seeing elongation factor G here, which also uses GTP, and it helps to, glide along the whole ribosome one codon. And when it slides along, it's gonna allow the tRNA molecules to remain bound, okay, to in that complementary fashion that they were originally to the mRNA, so that they remain, base paired to the original codon, but they're now going to be found in a different compartment within the ribosome. The ribosome moved relative to the two tRNA molecules, so they remained, intact in terms of their base pairing, but the ribosome itself moved forward. And so the result is that you're going to have the tRNA that was in the A site move into the P site, okay? And the tRNA that was in the P site now move into the E site, and that is what you're seeing in this bottom image here. Okay, now we're again positioned to allow an additional tRNA to enter the A site recognize the codon there and contribute and, the fourth amino acid to the growing polypeptide chain. Okay, so we'll start again from top and we'll go through, the recruitment of the correct tRNA to the transpeptidation reaction, which is the formation of the peptide bond, translocation, and then you will move on to the fifth codon. So these three steps will continue to occur until the ribosome has read all the codons. So if there are 90 codons, before the stop codon, then the ribosome is gonna repeat these processes 90 times until the 90 amino acids have been linked together via peptide bonds into a polypeptide. Now, the transpeptidation reaction that is carried out by 23S ribosomal RNA involves the tRNA molecules that happen to be in the P site and the one that is in the A site, remembering that the one that is in the P site is carrying with it the polypeptide. So the adenine that you have that is attached to generally a single amino acid would actually be attached to the polypeptide in the P site. Now, the tRNA, the incoming tRNA in the A site is going to use its amine group to react with the ester bond of the tRNA within the P site. And the result is that the tRNA in the P site would kind of hydrolyze that bond and kind of give up the polypeptide so that it's transferred to the tRNA that happens to be in the A site. So it's just a visual demonstration of what we already talked about. Now, termination of protein synthesis requires that ribosome arrive at one of the stop codons. In this case, we're seeing a UAA. And generally, just like you have elongation factors and you have initiation factors, there could be termination factors or release factors. So we have release factors 1, 2, and 3 that are going to be involved in correctly recognizing that this, in fact, is a signal to further stop synthesizing the protein. And, one last time GTP would be hydrolyzed, and you're going to have eventually the protein diffuse away, the whole complex fall apart, and initiation factor 3 again combined with the 30th ribosomal subunit to keep the two subunits apart. So they can come back together again and they can be recycled as long as this 30S subunit undergoes the initiation complex formation one more time, right? So this process of terminating protein synthesis can occur at any of the 3 stop codons that we refer to as the nonsense codons because they don't code for any amino acids. And there may be, several release factors that might be involved that help recognize each of those separate, codons or stop codons. And, you might have multiple release factors. You might have just a single release factor, depending upon the organisms that we're talking about. And of course, this entire process requires a lot of GTP to ensure the accuracy of this process. Even if you have a single amino acid that is incorrectly inserted, it can warrant the destruction of that protein. So you don't want to undergo this really expensive process to synthesize an inaccurate protein or a misfolded protein, which you would have to degrade. So cells ensure that they do it right the first time. Now, protein synthesis might be completed at this time, but you just have a, just the primary structure of the protein, which is just a chain of amino acids, and it needs to be folded in its correct form before it could begin to perform its function of, say, an enzyme or a transporter protein. And so in this case, what we notice is that the cells have a mechanism to prevent the misfolding of the proteins. Often folding is driven by the hydrophobic stretch of amino acids because generally they would be found hidden away from the hydrophilic environment of the cell. So as soon as these hydrophobic amino acids are synthesized, they start to hide themselves, and so they promote the folding of the protein spontaneously. Now, in order to prevent the spontaneous misfolding, generally we have a protein referred to as a trigger factor that binds to some of these hydrophobic stretches of the protein as it's extruding out of the ribosome until it's fully synthesized. And then just the trigger factor alone is able to help 70% of the proteins fully synthesize. Then there's the remaining 30% that need help from additional helpers that we refer to as chaperone proteins. In this case, we're seeing that not only that the trigger factor is helping, but you have chaperone proteins DNA-J and DNA-K that associate with this protein until it's fully synthesized, and then they use additional chaperones that we refer to as the GRIP-E in presence of ATP that would help 20% of that remaining 30% total to fold accurately, but you still have about 10% of the proteins that can't fold even just with the help of DNA, J, and K, and that is where additional these barrel-like chaperone proteins come in that we refer to as the GRO-EL and GRO-S, proteins that are kept linear using additional, chaperones like DNA, J, and K, would be inserted into this barrel. It generally has a cover, which is shown as GRO-ES, and somehow within this environment of the GRO-EL, the protein is able to properly fold in its three-dimensional shape. I'm sorry, I'm having trouble talking, but so it will eventually fold in its three-dimensional shape, and when it's released, it's properly functional, and you could use it for whatever purpose it was intended. So majority of the proteins would be properly folded without the help of chaperone proteins. 30% would require help from chaperone, and of that 30%, 20% would require just GRIP-E, whereas the remaining 10% would require GRO-EL and GRO-ES. Now, these molecular chaperones were initially recognized because their expression increased tremendously in cells that have been subjected to, say, acid shock, where they were taken maybe from a neutral environment and put into a slightly acidic environment, or heat shock, where they were taken from, say, their optimal temperature into a slightly elevated temperature. Because the reason of overexpression of these chaperones is because you were going to have these denatured proteins as a result of being exposed to slightly acidic conditions or slightly, hot conditions. And unless cell has a way of refolding those proteins back into their, what we refer to as their native form, their fully functional forms, cell would lose a huge amount of proteins. It might not have the energy to synthesize all of them at once, and might not have time to synthesize all of them at once. So it's sometimes easier to just take those misfolded proteins that, if possible, fully fold them back into their original form. So, these are not only synthesized. Now we know that these aren't only synthesized when we have that heat shock and when we have that acid shock situation going on. They will be synthesized normally to help some of the proteins fold properly. Finally, the last slide, where there are some proteins that are not going to be cytosolic. They're going to either be, say, excreted, say, within the outer membrane and the inner membrane, or just excreted into the environment. Or you could have proteins that are inserted into the plasma membrane. Okay, so there are mechanisms that help bacteria achieve that function either in conjunction with translation or post-translationally. So we have a couple of mechanisms here. One is post-translational translocation, where we're trying to insert a protein, say, into the membrane, or we're going to, at least first step is that you insert it into the membrane, and then depending upon whether it needs to be fully excreted, so it could maybe lose its association with the membrane, or keep it depending upon what the cell intends to do. So in post-translational translocation, you have a fully synthesized protein that will be inserted into the membrane. And so of course, this is going to require first differentiating this protein from cytosolic protein. So there has to be some way of figuring out that this protein actually belongs in the membrane and not within the cytosol. So there happens to be a sequence at the amino terminal of such proteins that we refer to as the signal sequence that is recognized. Okay, so in this case, we're seeing that the signal sequence is already associated with this channel that has formed. Okay, so there's this channel that has formed using the SEC-YEG protein that helps to secrete this fully synthesized protein, say from the cytosol into the external environment. But where does the physical force to shove this protein through this channel come from? So that force, that mechanism is going to be provided by SEC-A protein that is going to use ATP, okay, to sort of push this protein through and get it to where it needs to go. You can also have this co-translational translocation, where as soon as the signal sequence is synthesized, the entire this complex, this translational machinery is recruited. The exit site for the polypeptide is going to align with this channel, still made up of the same SEC-YEG protein. And it's going to, as the ribosome continues to synthesize the polypeptide is going to physically be pushed through the channel, okay? And of course, the cell can decide whether to keep this signal sequence or just cut it off, and then this protein would completely be extruded into the external environment. And then, once the translation is completed, the ribosome disassembles and this whole process sort of ends. So cells have these two mechanisms, two ways of taking either fully synthesized protein and translocating it to the outside of the cell using this channel, the SEC-YEG, or they could do it as the ribosome is synthesizing. So the energy that is being spent on the synthesis of the protein is going to be sufficient to push this protein through, of course, this SEC-YEG pore to get to the other side of the membrane. Of course, this process is a little bit more complex, and I didn't want to get into it just now. Hopefully, when you take cell biology, you'll also talk about how these proteins are weaved through the plasma membrane to, say, make a porine protein. And its process is going to be a little bit more complex for gram-negative bacteria that are going to have the inner membrane as well as the outer membrane, and they need to not only insert proteins into the inner membrane, they need to insert proteins into the outer membrane, right? Such as like porine proteins happen to be in the outer membrane, yet there are no ribosomes within the periplasmic space. So it happens to be a lot more complex, but at the very minimum, understand that there's going to be mechanisms involved beyond just the synthesis of the protein and just its, folding. It needs to get to a proper region as well. And there's going to be mechanisms that are going to help cells achieve that as well. I'm going to stop at this slide.